Please welcome Dr. George Amato.
Thanks, Sharon. Good afternoon. First, I'd like to thank Sharon and all the organizers of this very special symposium. It's really a pleasure to be here with you. I've prepared a little bit of talk. Barcoding is really near and dear to my heart, but after Sharon gave the introduction, I still think you should know who's speaking to you, because it influences, how we talk about things and the way we see the world. So I'm going to digress for just a moment to tell you a quick story. So when I was 10 years old, now that's when everyone groans, because it's like anytime someone begins a story with saying, when I was 10 years old, you should groan. But when I was 10 years old, I was watching a television program about a very famous paleoanthropologist named Louis Leakey. So this was a very long time ago, and there wasn't the internet, and so we got a lot of good information from the television. And there was a special, a National Geographic special, where Dr. Leakey had discovered this new hominid fossil in Olduvai Gorge in Tanzania called Zinjanthropus. Well, I was watching this, and I was looking at these fossils, and I was seeing the pictures of the Serengeti ecosystem around Olduvai Gorge, and I said, it's not too early to start looking for a job. And so I wrote a letter to Dr. Leakey and said that I would like to come work with him, when I'm a little older, and to study these fossils and to work with in Africa. And I also thought it would be a good idea to do some drawings for him, the way I imagined the ecosystem at the time of Zinjanthropus. And of course, again, there was not an internet, so basically the address I could come up with was Dr. Louis Leakey, Nairobi, Kenya. And I packaged all of this stuff with the help of my dad, into an envelope and sent it off. And 3 months later, I got a letter from Dr. Louis Leakey. And he said he was looking forward to me coming to work with him in Africa. And oh, by the way, thank you for the drawings. And I know I was 10 years old because the letter still hangs above my desk, 1966, so giving away my age. And I see that every day, and it means a great deal that he was such an important influence in encouraging me to study what I studied. And so I never miss an opportunity to come and talk to students who are just embarking on their science careers. I'm not sure why it's not advancing. Oh, here we go. So as Sharon mentioned, I came to the Museum of Natural History in 2005, and it was to organize a new institute at the Natural History Museum, our Institute for Comparative Genomics. The museum had been doing work in molecular biology for many years, but at that time, the museum decided we really needed to have an organized institute to really better define what we wanted to accomplish with the field of genomics at the Natural History Museum. We developed a mission that included such things as developing a comprehensive tree of life for all species, new methods in phylogenomics, a large repository of frozen tissues for research. One thing that was very interesting was we decided to develop a microbial research program as well. And so it was really we saw our purview as the world of the non-model organism in terms of genomics. And so if you come to visit at the museum, we have a number of large laboratories, a bioinformatics and computing center. But the one little part that I wanted to share behind the scenes with you is our molecular and microbial research collection. And the reason I wanted to share this with you is that this is really sort of a non-traditional collection for a museum. So many people are familiar with the fact that we have 33 million specimens, and that includes 2 million birds, skeletons, skins, fossils. But in this repository, we have the largest collection representing genetic diversity across the planet of any place in the world in those vats. And that is over 150,000 specimens, everything from microbes to skin biopsies from humpback whales, and so much of everything in between. Outside of there in our service yard is a 3,000-gallon tank of liquid nitrogen that's part of keeping that facility functioning and cold. And if we lost power for 3 months, that facility would be perfectly safe. This is sort of old-fashioned barcoding. All of our tubes here have a traditional barcode label, and we can access any one of our 150,000 specimens in just a few minutes. And we loan, as we call it loan, but we send out thousands of specimens every year to researchers all throughout the United States and around the world to be able to enhance their research. And one of the things that we've been doing as part of the research now is routinely barcoding, DNA barcoding, all of our samples. And so it's a really interesting place, and if you ever come visit me at the museum, I'd love to show you behind the scenes. The program within the Genomics Institute that's nearest and dearest to my heart is the one focused on the conservation of biodiversity. This is something that I've been working on for over 30 years. And the focus of the program is to use these technologies, similar to what you've all been doing in your projects, to both better understand threats to endangered species, but also to use that information and that research to ameliorate those threats and develop management programs. I was very lucky to be here on this important campus in 2003 at the Banbury Center for the very first DNA barcoding organizational meetings. It was a very exciting time to think about all coming together as a community of scientists across the world to think about what utility DNA barcoding might have, especially if it was done in a coordinated manner, developing large databases like BOLD, and as well as fine-tuning both our techniques and, again, this very large database. So that was in 2003, but it wasn't until 2005 when I came to the museum that I really began a very large DNA barcoding project at the museum. And today, I just wanted to briefly share with you 2 different ways that I've found DNA barcoding as an extraordinarily valuable tool for biodiversity conservation. The first one that I'm going to talk about is really for basic species IDs or binning of samples for other kinds of research projects, both in conservation genetics and molecular ecology. And then the second is DNA barcoding as a tool, a forensics tool, for monitoring the illegal trade in wildlife. You're all experts on DNA barcoding and also have benefited from the fact that this has been a very successful worldwide effort for many years. And that especially using a standardized small DNA fragment, again, in the animal world, primarily CO1 from the mitochondrial genome, we've just created this extraordinarily valuable database. So one project that I've had ongoing for a number of years has been focused on understanding the threats to large carnivores, especially the large felids. And this has been a collaboration with another major foundation here in the city of New York, the Panthera Foundation, which is all of its efforts are focused on the conservation of the world's large cats. So we've had projects on tigers, snow leopards, lions, jaguars, cheetahs, and a variety of other species. And so why are cats threatened? These large cats are threatened for many reasons, but primarily they're the direct killing of the animals, sometimes for body parts and for traditional medicines, other times due to human-wildlife conflict. Loss of prey in many areas where the prey of these large animals is being overhunted, then there's literally nothing for the large cats to eat. And of course, habitat loss and fragmentation. And the result of this has been devastating. So jaguars today only occur in 40% of their historic range. For lions, even though you can see them all the time on television, Nat Geo Wild or some program, they only occupy 18% of their historic range currently. Tigers, only 7%. And that's just over the last, since 75 years ago. We don't even know for snow leopards. So the focus of this program from my group has really been to sort of get a picture of the landscape of these animals, how they move around the landscape, how their populations are connected, or how they're being fragmented and disconnected. And we do all of this through non-invasive sampling. So in this little cartoon, we start with a jaguar, and then we have jaguar scat. And from the fecal material, we can extract DNA from that jaguar. And of course, now we can do everything from examining any part of its genome to answering some very practical kinds of questions. The advantage to this is that these animals are very difficult to see in the wild. They're really very shy. And even if you put camera traps or other kinds of devices out there, you don't get a very clear picture. But by using walking transects, using scat-sniffing dogs that help find these materials, we can literally collect hundreds and hundreds and hundreds of fecal samples. And of course, the great thing with these endangered species is without ever having to handle the animal. Now, of course, be careful what you wish for. So if over the course of a year we receive 5,000 scat samples, one of the first things we need to do is figure out which of those scat samples for sure are from our target species. So the first thing that we do after extracting DNA from these fecal samples is to barcode. And barcoding has proved to be tremendously valuable in that from 1,000 fecal samples in a national park, where we can find out that, out of those 1,000, 500 of them are jaguars. The other 500 are ocelots and a whole variety of other animals that may be useful for other people's research. But in terms of us answering the question about the jaguars, being able to barcode it and then focus on our target species is really important. And once we're able to do that, we can then genotype each individual. We used to do this with microsatellite markers, which you may be familiar with, and now we do it with a SNP panel. But we can identify it to the individual. So even though when we're out in the field we might not see a single jaguar, we can say, after recovering all these samples, that there are 35 jaguars in the park, and this one is Jaguar A. He's a male. He's the father of these other jaguars. And he's the one that left the park and went to the other reserve or didn't go to the other reserve. So we create this entire extraordinarily detailed map of what the animals are doing, who they are, whether any of the strategies are being successful in terms of allowing them to move between their corridors, and most importantly, when we're trying to connect these different fragmented parts, knowing whether or not they successfully were reproducing and passing their genes on. And so that's been, for us, has been extraordinarily important. And there's many, many examples of our molecular ecology work where we always begin with some form of barcoding to be able to bin our samples. It's true what we do with our environmental samples, or many other samples where you don't have the whole organism right in front of you. This is just a picture in the lab of the cat team, and of course, they're the ones who do all the hard work. But mostly what I wanted to talk with you about today was the second part. And with very generous and long-term funding, when I first came to the museum from the Alfred P. Sloan Foundation, we began a major DNA barcoding project that was focused on species that are threatened by commercial trade. So you may not be aware of this, but the illegal trade in wildlife, that's everything from products, specialty foods, exotic pets, a whole variety of reasons that people trade in endangered species and wildlife. That trade is estimated to be third only in terms of the amount of dollars than illegal drugs and illegal weapon sales. So estimated at more than $25 billion a year. We're really interested in better understanding the impact of this trade on endangered species, but also beginning to think about how barcoding might more effectively not only allow us to monitor this trade, but also to help with law enforcement. When we were beginning this back in maybe 2006 or 2007, Newsweek magazine at the time was just highlighting one of the issues of the illegal trade in wildlife, and it's what was called the Bushmeat Crisis. And it was as forests were being opened up for logging, there was an increase in hunting of wildlife. So instead of just being sustainable local hunting by local people, it was becoming commercialized, and forests were literally being emptied of their wildlife. So this is some photographs from Central African Republic, dwarf crocodile, other crocodilians, red river hogs, duiker antelopes, a whole variety of species were being emptied. And one of the groups of animals that was being heavily impacted were primates. And it was really putting a number of species in grave danger. So one of our first DNA barcoding studies was applying the barcode to see if it would be useful in identifying meat in markets. Because so often you didn't have the whole animal. You had pieces of meat and, or just a leg or something like that. And so we began an initial experiment to just examine many different species, but also to work on techniques, using different methods of sampling, different extraction methods, a whole variety of things. We did this in both looking at wild specimens in forests in a couple of different places in the world, but we also looked at museum specimens, seeing if we could match the barcodes that we were getting with the animals in the wild with vouchered museum specimens, since this was very important, especially to law enforcement, and actually just to better understand how much variation there would be in barcodes and the utility of it. In our very first study, this is just a large number of animals with their scientific names, but it ranges from crocodilians to antelope to monkeys to a whole variety of things. And we focused in the areas to begin with because there was a lot of bushmeat hunting in these various areas. We were able to focus in on a number of primate species so that we could get a good sense of just how sensitive the barcode would be for species identification. And then we published one of the first papers about barcoding, especially as it related to bushmeat, and basically demonstrated that it had strong utility. It was very accurate in describing species in almost every example, and that you could have just bits and pieces and you could get perfectly accurate identification of that product. But some other interesting things came from it as well, too. One was when we were focused on these dwarf crocodiles. So as we pointed out in this paper, our barcode, which only varied less than 2% in terms of sequence divergence within individuals within the same species, That worked for every species we looked at except for one. And that one species that it didn't seem to work for, that seemed to be really very divergent barcodes, was from this animal, the African dwarf crocodile. So we decided to try to understand, why was that? Was it something unique about dwarf crocodiles, or were we missing something? And we ended up doing a study examining something like 600 or 1,000 dwarf crocodiles across all of their range. And what we discovered and published was that the reason the barcode didn't work when we just applied it to a bunch of different samples was that there are actually 3 diagnosably distinct species of dwarf crocodile in different parts of Central Africa, and that when we did our first study, we had just arbitrarily looked at different samples. Once we went back and did a detailed analysis, and not just barcoding, but obviously looking at nuclear DNA sequences and a lot more data, we were able to show that this was some cryptic diversity in Central Africa that had been previously unrecognized. And that was a very exciting discovery for us. And so it's another way that we've used DNA barcoding, where we can go in and rapidly barcode a lot of species. But when you get, if you're barcoding 2 frogs from Borneo, and they look kind of alike, but their barcodes are really different, that's a good indication that perhaps you should investigate that further, and that you may be looking at cryptic diversity. And so that was a really good example of that. Now, I mentioned bushmeat was the first thing we did, but one of the things that I felt very sensitive about our work on bushmeat was that bushmeat sounded very much like we were studying this problem that existed over there, some other place. Those people are eating bushmeat, and we're going to help stop that. And I knew that we eat bushmeat here. It's just we call it seafood. So seafood, a lot of seafood is bushmeat. It's wildlife that we harvest. Some of it sustainably, some of it unsustainably. And so I felt very strongly that I wanted to do a barcoding project that was focused on our bushmeat, essentially, here right in New York. And so we began a project, and I had a student working with me on this project, and we wanted to develop the barcodes for every species of tuna that people consume. And one of the reasons that we wanted to focus on tuna was that there was a proposal, finally, for the first time, to regulate the harvesting of, by CITES, the Convention on International Trade in Endangered Species, a particular species of fish, and that was bluefin tuna. And the reason for this was that bluefin tuna had been basically over-harvested until 95% of bluefin stocks had disappeared. So we were able to develop barcodes for all of these species of tuna by literally going out and getting fish from fishing boats around the world, vouchered specimens that we knew that what this fish was for certain. We developed the barcodes, but then we did something that then has become a very popular thing, which is we decided to barcode tuna in our favorite sushi restaurants. So this was a novel thing back then, and especially I think the student who was working with me, Jake Lowenstein, at the time, said, when I came to him and said, how about going to have lunch at 30 sushi restaurants? I'll pay the bill, and you go to every one from the one right around the corner from the museum that has a lunch special. I'm not sure if that's how you should pick your sushi restaurant. But to the most expensive sushi restaurant I knew of at the time that was in the Time Warner building. And Jake went and ordered lunch. He ordered a lot of tuna, and of course he brought it back, little bits of it back to the lab. People must have been staring at him in the restaurant. Well, he's in these fancy restaurants and then putting a little bit in a microfuge tube. But the thing that we discovered was that it was misidentified all over the place. And everywhere it was misidentified. And I'm not suggesting that the tuna wasn't fresh or delicious. It's just somewhere between being caught, sold, wholesale, and all of these things, it was incredible how the level of misidentification. So on one hand, I'm thinking, well, okay, we're going to publish this in PLOS One, and I hope it's useful to someone. 4 days after it was published, the editors of PLOS One called me up and said the paper had been downloaded 14,000 times. They seemed shocked. I was shocked. And on one hand, I'm thinking, we just did a barcoding project. But of course, it was a big example of the value that barcoding can play in something like food safety, fraud, all kinds of other issues. So we had nice coverage on the front page of the Wall Street Journal and the New York Times. And the next thing that happened was I got a call from Washington, D.C., where the head of all of the laboratories for US Customs and Border Protection asked me to come down and give a seminar about barcoding. And out of that, we ended up doing training on barcoding at the museum for the 8 laboratory heads for the labs for US Customs and Border Protection, which was quite interesting. It was quite interesting to see what they were doing before this as their species identification tool. They were using a technique. I was the only one old enough in the lab who actually remembered the technique. Pre-PCR days. And so that was the beginning of a major partnership that we developed with a number of these folks. And that led to many other projects, including on endangered parrots, many other endangered species. We did a follow-up to caviar barcoding. A colleague of mine had done a sort of a version of barcoding a long time ago on caviar, and after the sturgeon were protected in the trade, it seemed a good idea to once again go ahead and barcode a lot of caviar. We did that and were able to show how the regulations had really affected and reduced the amount of trade in endangered species. We were then asked to expand into things that were non-traditional kinds of things to barcode. Partially or fully processed leather products, food supplements, and a whole variety of other things. For a while in the early days, it was just so much fun. It was like, what can you go to your refrigerator and barcode? Can you barcode that if it's been cooked and canned and processed? I don't have this, but my favorite was someone came in with a bag of dog biscuits made out of bison. That's what the bag said. And so I'm thinking, well, okay, we haven't sequenced any bison in this lab. It's no contamination issue. So let's give it a try and see if we can get a fragment of a barcode of a bison. Presumably the meat was cooked, made into some kind of a biscuit, baked, and we were able to get bison barcodes. We call them mini barcodes, where we made them slightly smaller fragments than the traditional barcode and pieced them together. But it was kind of a fun time to be just seeing how far you can do that. I think everyone felt like they were some food investigator. But also what we were able to do was produce training materials, tools, barcoding manuals, and we worked with a very large number of folks. This work continues today, sometimes with barcoding, sometimes with what we call barcoding plus. And I just wanted to share with you 2 images as I finish up here about some more recent kinds of cases. I'm happy to share with you what must look like a very odd photo. So we, for a long time, were barcoding ivory. And the reason we were barcoding ivory was the fact that the laws governing the sale of ivory varied based on whether it was an Asian elephant, an African elephant, a savanna elephant, a forest elephant. And so we would actually have to barcode the ivory. It's not a trivial process. It works, but it's not trivial. Luckily, the law changed 2 years ago, and it in the state of New York, it's illegal to buy, trade, or sell any ivory. Any ivory. Any elephant, mammoth, any ivory. And so now it's much easier. It's not so hard to identify ivory. I've made experts of all the local New York State DEC detectives who are friends of mine and who were standing on the roof here the night after I went with them, and it was exactly 2 years ago now. I got a call saying, meet me on the corner of West 57th Street, and we're going to go in and do a raid on this high-end store. And there was $4 million worth of ivory in the showrooms upstairs. So I'll tell you, it was a rather interesting day. And at the end of the day, the detectives posed with some of the objects. And all of this has been successfully prosecuted, and it was also an interesting day testifying in front of the grand jury on ivory. Even more recently, we've been working on rhino horn smuggling. That's an extraordinarily large rhino horn that some US Fish and Wildlife folks brought to me not too long ago for a federal case here. But the one that I wanted to share with you, which was not this case, but which was more related to what we're talking about today, is they brought this object. It's called a libation cup, and it's supposed to be carved out of rhino horn. But again, to be able to prosecute someone who will then, they'll be selling it as rhino horn for, I think this cup was selling for $100,000. We have to be able to actually identify it. And we were able, under the bottom of it, we scraped into it, and we were able to barcode it and show unambiguously that this was from a black rhinoceros, and this case was successfully prosecuted. So using barcoding in forensics applications in the wildlife trade has allowed us to partner with all of these various agencies, and I think to really make a difference in both how we monitor the trade and how we enforce the laws. And that's been a very rewarding exercise. And so while most of our research is very basically trying to understand the diversity of life on the planet at the museum, and it's very basic evolutionary research, there are these realms, and barcoding has turned out to be one of the realms where we can both do some interesting science, but also connect it to solving problems that are very practical problems to society. And when those areas intersect, it just can be very, very rewarding. So with that, I'd just like to acknowledge the various foundations and a number of individuals who have made all of this work possible. And thank you all for listening and encouraging you to continue in on your science. Take some questions.
All right, do we have questions for Dr. Amato? We have time for about a few questions.
Hello. So on cryptic diversification, which based on your slides looked like diversification at the species level, which you could then group into subcategories. What is the difference between that cryptic diversity and just the creation of a new species?
So how we categorize the world, as you know, is a very interesting element of biology, including how we define a species for sexually reproducing organisms. There's a very big body of literature for that. So you're right, we know diversity exists at every hierarchical level, right? There's only one species in this room, and yet there's a lot of diversity. And then as you look above that, at the species level, it's always been very important, that level, to biologists, because we believe that's the evolutionary unit in sexually reproducing organisms. And so we think that a species has a shared evolutionary trajectory that's different from others. And that's independent of higher level classifications. And so we use a phylogenetic species concept definition, and that's a definition that basically if you have a hypothesis, I think this is one species or I think this is two species, you can use a methodology examining for diagnostic characters, which is diversity that all individuals of a group share, but don't share with individuals from another group. And those are the characters that we look for to say, not just is this crocodile different from the crocodile next to it, but is this, all the crocodiles in this area, do they share a set of diagnostic characters that distinguish them from all the other similar crocodiles in another area? And we define it that way because we believe the only way you could have that pattern is if they're reproductively isolated and have been so for a sufficiently long period of time. And so that's the criteria that we use for saying, oh, there are 3 species here, not one species with a bunch of diversity. Longer answer, probably, than you wanted. But usually that's a seminar, like, for a whole semester. So I just tried to get that in 2 minutes. There's a question here.
Is this, the data, is it available for student access through the American Museum of Natural History?
Sure. So, I mean, everything that I've talked about actually represents many publications. So all the publications are available, and then the publications will link you. Primarily, all the sequences are deposited both in, are all in GenBank. Some are in Bold and GenBank. But everything that we do is, in fact, it's a requirement by most scientific journals now before your paper, when your paper's out for review. So yes, all the data are available. And I'm happy to point anyone in a direction that they have a particular interest in. But obviously, GenBank is where to begin your searches. Recognizing that BLAST is not always the best way to find things. So that's another semester-long discussion. Any other?
Other questions?
Great.
All right, let's thank our keynote speaker again.